Welcome to another episode from the Veterinary Medicine Digest. Today we are embarking on a deep and comprehensive journey into one of the most visible yet misunderstood phenomena in animal welfare and veterinary science, the development of stereotypies. These repetitive, unvarying, and apparently functionless behaviors are not just odd habits, they are windows into the psychological and neurological states of the animals under our care. Worldwide, it is estimated that over 85 million animals spanning farms, laboratories, and zoos exhibit these behaviors, which suggests a staggering scale of potential welfare compromise. To truly understand what is happening when a sow bites her bars or a mouse flips incessantly in its cage, we must look beyond the surface level of behavior and explore the complex interplay between environmental confinement, physiological stress, and the very architecture of the brain. To set the stage for our discussion, we must first look at how these behaviors manifest in a real-world setting, specifically within the context of intensive pig production. One of our foundational sources, a study conducted at the Northeast Agricultural University, provides a detailed look at how confinement duration and age impact the welfare of pregnant sows. The researchers focused on Sinman pigs, specifically the large white genetic line, looking at 50 sows across different parities, from those in their first pregnancy to seasoned veterans in their fifth. These animals were transitioned from relatively spacious pens into gestation crates that measured 200 centimeters by 65 centimeters, a space that essentially prevents them from turning around or investigating their surroundings. In this environment, the sows began to exhibit a range of what we call oral stereotypies. This includes bar biting, which is defined as nosing, rubbing, licking, or biting any metal component of the stall other than the feeding pipe. There is also trough biting, where the animal focuses its repetitive energy on the feeding trough itself, and vacuum chewing, where the sow engages in continuous chewing motions even though no food is present in her mouth. The research showed that while young sows might not show a significant increase in these behaviors during the first 8 days of confinement, the cumulative effect of spatial restriction becomes undeniable as time progresses. As the confinement duration increased from 25 to 100 days of gestation, the frequency of vacuum chewing and bar biting rose significantly across all groups. This suggests that the negative effects of a restrictive environment are not just immediate but cumulative. The longer the duration of the stressor, the more ingrained and frequent the abnormal behavior becomes. Parity, or the number of times a sow is given birth, also played a crucial role. Older sows, those in their fifth parity, showed significantly higher frequencies of vacuum chewing and bar biting compared to younger animals throughout the entire pregnancy. This supports the idea that once these behaviors are established, they become more severe with increasing age and are incredibly difficult to eliminate. But the behavior is only half of the story. The physiological data from this study provides a striking look at what is happening inside the bodies of these sows. As the duration of confinement increased, the researchers observed a significant rise in serum cortisol, which is often used as a primary indicator of stress. Alongside cortisol, there were marked increases in pro-inflammatory markers like interleukin-6 and immune indicators such as immunoglobulin A. The older sows consistently showed higher levels of these stress and inflammatory markers than their younger counterparts. Interestingly, the study also monitored acute phase proteins like C-reactive protein and pig major acute phase protein. In all but the first and second parity groups, these proteins increased significantly during the confinement period. However, there was a fascinating shift when the sows were moved to the farrowing beds on the prenatal day. Their serum cortisol, immunoglobulin A, and interleukin-6 levels, as well as the frequencies of bar biting and trough biting, decreased significantly. This suggests that even a slight improvement in the environment, providing the sow with a bit more comfort as she prepares for delivery, can temporarily relieve the psychological and physiological pressure. This leads us to a broader question that has debated for decades: is stereotypy a reliable indicator of poor welfare? Georgia Mason and N.R. Latham have explored this paradox extensively, noting that while stereotypies are born from negative environments, the relationship between the behavior and the animal's current suffering is not always straightforward. They suggest that we need to look at 4 distinct processes that might obscure the link between a repetitive behavior and current welfare: do-it-yourself enrichment, mantra effects, central control as a habit, and perseveration. The concept of do-it-yourself enrichment posits that some stereotypies might actually help the animal cope with its environment. For instance, certain oral movements in cattle generate saliva, which can act as as a natural buffer against stomach acid, a major risk for animals fed on concentrate diets. In this sense, the behavior might be a functional replacement for a natural foraging activity that is being denied. Then there is the mantra effect, where the sheer rhythmicity and repetition of the behavior might induce a state of tranquility, much like chanting or rhythmic rocking in humans. If a behavior provides a calming effect, an animal performing it might actually have better welfare than a non-stereotyper in the same barren environment who has simply succumbed to a state of apathy or depression. However, as behaviors are repeated thousands of times, they can transition into habits controlled by different parts of the brain. This is known as central control, where the behavior becomes an automatic process that requires minimal cognitive processing or sensory feedback. Once a stereotypy reaches this stage, it can persist long after the original environmental stressor has been removed, making it a poor indicator of the animal's current state. Finally, there is perseveration, which is a broader symptom of altered behavioral control. Animals in highly restrictive environments can develop central nervous system dysfunctions that impair their ability to regulate any behavior properly, leading to a general inability to switch between different activities or respond appropriately to new stimuli. These long-term consequences of confinement do not just affect the individual animal, they can be passed down to the next generation. Research reviewed by Patricia Tatemoto and her colleagues highlights how the prenatal environment can shape the development of the fetus. This is often explained by the thrifty phenotype hypothesis, where the maternal environment provides signals to the fetus to help it prepare for the postnatal world. If a mother is chronically stressed, she may pass on harmful cues to her offspring, affecting their emotional reactivity, social behavior, and responsiveness to stressors. Interestingly, there is evidence that the specific behavior of the mother matters. Sows that exhibit high levels of sham chewing or other oral stereotypies during pregnancy have been found to have offspring with decreased fear responses. This might be because the mothers who are actively trying to cope through stereotypy are in a slightly better psychological state than those who have given up, thereby providing a more organized environment for fetal development. The mechanism behind this transgenerational shift is likely epigenetic. Stress can cause changes in DNA methylation patterns within the limbic system of the brain, specifically in the hippocampus and amygdala, which can permanently alter how the offspring perceives and reacts to the world. This brings us to our main featured article by Jinyu Zhang and his team, published just this year. They argue that we must shift our perspective and recognize stereotypies not just as behavioral abnormalities, but as behavioral markers of fundamental brain dysfunction. They contend that these behaviors are manifestations of impaired neuroplasticity and convergent dysfunction within specific brain loops, specifically the corticostriatal thalamocortical circuits. These circuits are the brain's command center for behavioral selection and inhibitory control. They balance our goal-directed actions, things we do with a purpose, with our habitual actions, things we do on autopilot. In a healthy brain, the prefrontal cortex exerts top-down regulation over the striatum, allowing for flexible behavior that changes based on environmental feedback. However, under conditions of chronic stress or extreme environmental deprivation, this regulation is compromised. The brain begins to favor inflexible, habit-dominated patterns. Behavior becomes automatic, repetitive, and increasingly insensitive to the world around it. Zhang and his colleagues describe stereotype behavior as a cross-species neurobehavioral phenotype. Whether it is repetitive jumping in a deer mouse, root tracing in a laboratory mouse, or bar biting in a pregnant sow, the underlying neurobiology is remarkably consistent. These models all show signs of dopaminergic dysregulation, where the brain's reward and habit formation pathways are overactive. The transition from a flexible action to a rigid habit is facilitated by dopamine signaling in the striatum. This is why pharmacological agents that block dopamine receptors, such as haloperidol, can significantly reduce the frequency of established stereotypies. The researchers also point toward a molecular signature for this transition, a transcription factor called delta FOSB. Delta FOSB is known to accumulate in the striatum following repeated neural activation, and it is a hallmark of both substance-related and behavioral addictions. Its presence is associated with compulsive behavior and a marked decrease in behavioral flexibility. Zhang proposes that elevated delta FOSB expression might be the molecular signature of long-term stereotypy, effectively making the behavior a form of behavioral addiction. At the circuit level, these behaviors are further stabilized by structural changes in the brain. Chronic adverse conditions can lead to maladaptive forms of neuroplasticity, including alterations in glutamatergic transmission and the remodeling of dendritic spines. These structural changes effectively lock the brain into these maladaptive habit circuits, making them incredibly resistant to change even when the environment improves. This is why simply adding a few toys or a bit of straw to a barren cage often fails to stop a well-established stereotypy. The brain itself has been physically altered. This neurobiological perspective has profound implications for how we assess and treat animal welfare. it suggests that conventional environmental enrichment, while essential, may not be enough to reverse established brain dysfunction. Zhang and his team advocate for more targeted interventions. First, we must prioritize early life environmental complexity. Providing young animals with diverse and challenging environments during critical developmental windows can help preserve their neuroplasticity and prevent the consolidation of these rigid habit circuits in the first place. Second, we might need to explore combined environmental and pharmacological strategies. By using medications that target the dopaminergic or glutamatergic systems in conjunction with environmental improvements, we might be able to facilitate a rebalancing of the brain's circuitry and enhance the reversibility of long-standing behaviors. Third, our frameworks for assessing animal welfare need to evolve. Relying solely on behavioral observations can be misleading, as we have seen that the absence of behavior doesn't always mean the absence of suffering, and the presence of behavior doesn't always reflect current distress. Future welfare assessments should incorporate neurobiological biomarkers. This could include measuring behavioral flexibility through specialized tasks, using neuroimaging to assess connectivity between the prefrontal cortex and the striatum, or even looking for molecular indicators of maladaptive plasticity like delta phosphate. In conclusion, the journey from a barren gestation crate to the complex neural circuitry of the brain reveals a deep and troubling story of how we manage the animals in our care. Stereotypies are not just annoying behaviors to be suppressed through physical restraints or hot-tasting substances, they are a cry for help from a brain that is struggling to adapt to an inadequate world. When an animal is forced into a life where it has no control, where its basic instincts are frustrated at every turn, the brain eventually breaks. It rewires itself to survive, but in doing so it loses the very flexibility and adaptive capacity that define a healthy life. As veterinary professionals and researchers, our goal must be to move beyond reactive symptom management. We cannot simply wait for a behavior to appear and then try to fix it. We must proactively preserve neural function by providing environments that meet the complex biological and psychological needs of every species. Whether it is a sow on a farm, a mouse in a lab, or a polar bear in a zoo, every animal deserves a world that allows its mind to stay inspired and its brain to stay healthy. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging.